Welcome back to our third episode of sexual reproduction. Today we're going to look at sexual reproduction in plants. As per our outline, we are going to look at floral structure, and I'm going to make a different video for that. We are going to go through gametogenesis in plants in this video. We shall look at pollination, double fertilization in plants, and I will make a different video for alternation of generations. So through this course outline we are having, we shall make 3 videos out of it. Alternation of generations will be different and floral structure will be different. However, today we are going to emphasize gametogenesis in plants, pollination, and double fertilization. When we proceed, I would like to request my students never to give up, and I will emphasize, be like that frog. Before it's swallowed, at least it grips the neck to prevent its swallowing. However, gametogenesis in plants, we are going to begin with the definition of gametogenesis, which is the process by which gametes are produced. In our previous episodes, we've been looking at asexual reproduction, but this one introduced us to sexual reproduction, where reproduction occurs by fusion of gametes. In the process of formation of these gametes is what we call gametogenesis. I think when you try to compare with the other mode of reproduction, we are seeing a big difference. In plants specifically, gametes are going to be ovules or the pollen grains. The pollen grains are going to, if you're looking at this cursor, they are going to be formed in the anther heads, and the ovules are going to be formed in this part called the ovary. I think you're seeing that on our cursor. So within this process of gametogenesis, we are going to divide it into 2, formation of pollen grains and formation of the embryo sac and ovules. And we are going to first look at the process of development of the pollen grains. These ones, like I've told you, they develop within the anther heads, and within each anther head, we have 4 pollen sacs. And these pollen sacs, each has diploid pollen mother cells. Within the diploid mother cells, this is where we develop the pollen grains. And how does this take place? It's all going to begin with the division of this pollen mother cell by meiosis to form a tetrad. A tetrad is simply a group of 4 haploid daughter cells. This tetrad is going to be rounded off, and rounding off simply means secretion of walls forming microspores. These walls are going to be cellulose cell walls, because this is a characteristic of all plant cells. Within the tetrad, there is only a single nucleus, and this nucleus is going to divide by mitosis to form 2 different nuclei. These include a generative nucleus plus a pollen tube nucleus. So after that happens, the wall of our pollen grain thickens to form an exine, which is an outer layer, and the inner layer is going to be called an intine. So eventually we shall come up with a haploid mature pollen grain. We've seen that in the formation, we have 2 processes of cell division. We have mitosis, which results into formation of a haploid tetrad, and then we have mitosis, which splits the nucleus into 2, into the generative nucleus and the pollen tube nucleus. So when we look at the entire process by virtue of images, you will see that our pollen mother cell, which is diploid, is going to divide by mitosis, both mitosis 1 and 2, to form a tetrad. This tetrad contains 4 haploid daughter cells. These daughter cells are going to be separated by secretion of walls, as we are seeing here, to become individual daughter cells. These walls are going to thicken to form a thick outer layer known as the exine, and it will form a thin inner layer, which is going to be called the intine. Through a process of mitosis, the nuclei in each of the walls, the nucleus in each of the tetrad cells, is going to be split into 2. One is going to be the generative nucleus, and 2 is going to be the pollen tube nucleus, as we are seeing. So when you are requested to describe the formation of pollen grains, you are very free to draw this kind of cycle, as long as you use what we call descriptive labeling. So in your drawing of this cycle, make sure there is an arrow that connects the individual haploid microspores from the tetrad to the mature pollen grain. So there should be an arrow that connects from here to the mature pollen grain. However much, we've not shown that arrow on our drawing here. So as a male gamete in plants, we look at its adaptations that make it survive to allow fertilization to take place. So among the adaptations of pollen grains, we have that thick exine, and this one protects the nuclei against harsh conditions. These pollen grains are light, therefore they are easily dispersed when released. You remember that dispersal can be by insect, by wind, or by animal generally. So dispersion is very crucial for fertilization to take place. The outer layer called the exine has a waterproof sporopollen, and this one prevents water loss, prevents desiccation. We have a pollen tube nucleus that germinates into a pollen tube to transfer the generative nucleus to the ovary to allow fertilization to take place, as we shall see during double fertilization. There is a generative nucleus. This one stores the genetic material. So that word, we apologize for that slight mistake. The entire pollen grain has very many wall layers. You remember we've talked about the exine, we've talked about the intine. And this one prevents germination within the anther head when the pollen grain comes into contact with water. Generally, pollen grains act as bioindicators away from the adaptations. These pollen grains act as bioindicators. So if we are told to explain how they act as bioindicators, their presence indicates less Mann effect. Therefore, it shows that there is absence of forest clearance. It shows that in an area, in a particular area, presence of pollen grains indicates presence of very many forests, and it shows less human effect in terms of deforestation, in terms of forest clearance, among others. That means that their absence shows rampant deforestation, because these pollen grains should come from plants anyway. So if you clear away plants, that means you're going to face a lot of challenges. So because of that, we are going to compare pollen grains and asexual spores we talked about during asexual reproduction. You remember in asexual reproduction, we talked about spores being a mode of reproduction commonly used by the fungi. So when we tell you to compare, we are looking for both similarities and differences. And among the similarities, both are formed by meiosis, both are haploid, both are covered by an outer layer called the exine, both are produced in large quantities, both are nucleated and are capable of germinating as long as they find an appropriate stratum. Among the differences, we can talk about spores are smaller, however pollen grains are bigger. That's why pollen grains, we tend to call them macro spores. Number two, spores are only formed by one process, which is meiosis. However, pollen grains can be formed by both meiosis and mitosis. Formed in the sporangium for spores, but pollen grains are formed in the anther heads. Spores germinate on their own. However, pollen grains can only germinate on an appropriate stigma, on a compatible stigma. So that also shows a big difference. Spores are only dispersed by wind, but we look at pollen grains being able to be dispersed by both wind and animals. Spores have only one nucleus, but pollen grains have two nuclei, i.e. the generative nucleus and the pollen tube nucleus, as we've seen in the structure. Spores can remain dormant for a longer period of time. However, when it comes to pollen grains, they can only be dormant for a shorter period of time. So among the differences between these two, I think we have displayed them very, very well. When we are requested to describe how an embryo sac and ovules are going to be formed, our process can be described very well by the images you're seeing, that cycle. It all starts with a diploid mother cell within the embryo sac. It's found within the ovary. This one is going to divide by mitosis. to form 4 haploid cells. These 4 haploid daughter cells, 3 of them degenerate, they do not develop, and only one is going to grow into a megaspore or a haploid embryo sac. This one is going to continue the process of growing because we are going to nourish the entire cell with nutrients being obtained from the nucleus to form the end nearest micropyle. This one, its nucleus undergoes division by mitosis to form 2 daughter nuclei within the cell, and it forms what we call the female gametophyte or the embryo. It undergoes repeated mitosis until when we form a cell of this structure, which we call an immature embryo sac, where we have cells we call synagids at one end, the cells we call the antipodal cells at the opposite end, and the 2 nuclei which we call the polar nuclei. So a process of nuclear fission occurs where these 2 polar nuclei fuse to form only one diploid polar nucleus, which is at the center of the embryo sac. So in a mature embryo sac, we have the synagids, the antipodal cells at one end. We have the synagids at another end. We have the diploid polar nucleus. Among these synagids, only 2 are synagids, and one at the center forms the female gamete, or what we call the ovum. I think you're clearly seeing this. So in a mature embryo sac, we have 2 synagids, 1 mature female gamete. We have 3 antipodal cells and only 1 diploid polar nucleus. So simply that is the structure in terms of images. However, let us look at the structure if we are requested only to write without drawing to see how best we can discuss our structure here. When we are requested to just write and describe the process we have previously seen, we discussed that the entire process begins within the ovary, where we have the diploid embryo sac mother cells. These ones divide by meiosis to form 4 haploid daughter cells. 3 of them degenerate, leaving only 1 to develop into the embryo sac. This embryo sac is going to become vacuolated and enlarges rapidly through a process of growth. The nucleus of this immature embryo sac undergoes successive mitotic divisions, forming 8 haploid nuclei at each end of the embryo sac. 2 of these 8 nuclei are going to migrate to the center to form the polar nuclei. And eventually, through the process of nuclear fission, the polar nuclei fuse to form a diploid polar nucleus, which is at the center of the embryo sac. The remaining 6, they are going to migrate. 3 of them migrate to different ends. Each will migrate to a different pole, as we've seen. When you look at the remaining 6, they are going to migrate to opposite poles. 3 of the 6 are going to separate at each end of the poles of the embryo sac. And these ones are going to become separated by thin cell walls, but remain at each end. Each end will have 3 cells, each separated by a thin cell wall. One of these cells at the micropyle end is going to become the female gamete, and it's going to be the haploid female gamete. Upon fertilization, it will form the zygote or the embryo. And the remaining 2 at the same end, where the female gamete is, form what we call the synagids. These synagids, I think we looked at them. and these ones, they are non-functional, so they will degenerate. We don't need them upon fertilization. At the other end, far away from the micropyle end, we are going to have 3 cells which form the antipodal cells, and also these cells do not have any function in fertilization. So likewise, when you compare formation of pollen grains and formation of ovules and embryo sac, you see to it that formation of ovules and pollen sac looks to be a little more complicated than formation of pollen grains. So in this, I would like to say that female organisms look to be more complicated than the male organisms, and their processes, or the processes happening in female organisms, are more complicated than processes happening in the male organisms. They are longer than the processes happening in male organisms, as we have looked at in terms of formation of pollen grains and formation of ovules and the embryo sac. So if you have really got the 2 processes, I would like to end this session by giving you some sample questions. For example, if you are told to outline the events that lead to formation of the following in a flowering plant, how can we go about it? And Roman 1, which is carrying about 6 marks, how can we form the events that form a pollen grain? How can we describe it from what I have discussed? In Roman 2, which can carry about 8 marks, how can we form an ovule and the embryo sac when we are discussing the same process? And I would like to urge you members that do not stop when you are tired, stop when you are done. So after watching my video, please try to go through the question I've given and make sure at the end you assess yourself. After trying out, you can send the answers via my email or my WhatsApp number, which I will repeat, 0705 533901. So you can send your answers through that channel such that we can respond to some of them. Part B of this very question, I would like you to research about how development of the ovule and the embryo sac in a flowering plant differs from oogenesis in humans. Here I would like my learners to research about that question and give me feedback. The second question I would like us to look at is describe the structure of an anther of a flowering plant. Here the solution is just so simple. You go draw the transverse section of an anther head. We looked at it in one of our discussions. You just draw that, label it very well, use descriptive labeling, and you will have the answers marked. Next, describe the structure of the stamen. We all know the stamen is the male part of the flower, and it is made up of filaments which support the anther heads. These filaments are elongated, enlarged at the base and taper anteriorly. These filaments hold bilobed anthers which are bounded with an epidermis. The lobes are divided by a suture along which they split on drying. So the anther heads contain numerous diploid mother cells which develop into pollen grains on maturing. So that is how you can respond to such a question. It has 5 marks, and simply we can accumulate the 5 marks by those few points. I would like to stop here and request you to share this video share my link with other members out there. Urge them to subscribe so that we can continue receiving this information and build up the proper understanding of reproduction topic. I wish you the best and thank you for attending to me. So we look at the structure of the organ where pollen grains are formed, and this is the anther. So when we are looking at the structure, we shall look at the transverse section of the anther, and I have organized this in form of a drawing. If we get our anther, just like you're seeing from our cursor here, and we make a transverse section, we split this anther head into 2 in a transverse section. What do we see when we apply a microscope to observe whatever is inside the pollen, the anther head? So we shall see our anther head in such a kind of structure where we show 4 different pollen sacs, and within these pollen sacs we have inside there a pollen mother cell. It is shown by this red color. Within the pollen mother cell we have the pollen grains, those individual round structures, which are 4. And within there we find a tapetum. There is a fibrous layer and the outer wall of the anther head called the epidermis. So generally, when you are requested to describe the structure of an anther head, you simply draw this. After drawing, you name each and every part. The central part is the vesicular band of the filament, which brings in water and mineral salts. These curvatures, these are lines of weaknesses, or you can call them sutures, where our anther head will split on drying to release the pollen grains. So simply, that is the structure of an anther head, and that is what we have to describe on that particular aspect.